If you look at the latest high-resolution electron microscopy of a human cell, the images just, I mean, they completely shatter those classic diagrams we all grew up with.
Oh, absolutely. The textbook models are totally obsolete now.
Right. You know, the ones where the nucleus and the ER and the mitochondria are drawn as these, like, isolated islands just floating independently in this vast empty jelly.
Yeah, it's just the static soup. But the reality, especially captured in this groundbreaking 2026 review article from Cell Death and Disease, it paints a radically different picture.
Exactly. And that's our mission for this deep dive today, to really change how you, the listener, visualize the inside of a cell. Because it's not a soup. It's more like a densely packed, physically wired metropolis.
Yeah, these organelles aren't isolated at all. They're actually physically touching, right? They're communicating and trading resources through these microscopic bridges called membrane contact sites, or MCSs.
And realizing that these physical contact sites exist, I mean, it fundamentally rewrites our understanding of cellular biology. We're looking at a highly synchronized infrastructure grid.
And what this review paper so brilliantly details is how cancer exploits this exact infrastructure. I mean, a tumor doesn't just rely on genetic mutation.
It actively hijacks these physical bridges.
Sure.
Right. To fuel its own survival and resist therapies.
Exactly. It hotwires the city's power grid to spread throughout the body.
Which is just a fascinating analogy. So today we're going to map out this cellular city. We'll explore, the specific molecular bridges being hijacked, how they bypass our best drugs, and how we might collapse that mutated infrastructure.
It's a lot to cover, but it's incredible science.
Let's start right at the center of the grid, looking at the most heavily studied physical connection in the cell, which is mitochondria-associated membranes, or MRRCs.
Right, so this is where the endoplasmic reticulum, the ER, physically docks with the mitochondria.
And the ER is basically this massive calcium reservoir, right?
Yes, exactly. The ER holds the calcium, and the mitochondria are the energy generators. So when they dock, they form this highly specific calcium funnel.
So like a literal water pipe between two buildings.
That's a perfect way to picture it. On the ER side, you have a release valve called IP3R. And then on the mitochondrial side, there's a receiving channel called VDAC.
Okay, so IP3R is the valve, VDAC is the receiver.
Right. And bridging the physical gap between them is this tethering molecule known as GRP75.
I love the acronym for this, by the way. A great way for you listening to lock this into memory is the acronym GIV. GRP75, IP3R, VDAC.
Yeah, they literally give calcium from the ER straight into the mitochondria.
But, if we think about calcium as the voltage for this power grid, why does cancer need to manipulate this specific connection? Like, what's the benefit?
Well, because calcium dictates the metabolic rate. Inside the mitochondria, an influx of calcium rapidly accelerates the TCA cycle.
That's the part that cranks out the energy, right?
Exactly. It cranks out massive amounts of ATP, which is the energy currency of the cell. And a cancer cell dividing uncontrollably, it requires an astronomical amount of ATP.
So it needs that calcium valve jammed wide open.
It does, but, and it operates on this terrifying metabolic tightrope. It's what we call the Goldilocks dilemma of oncology.
Because if the voltage is too low, the cancer starves. But if the voltage is too high, it blows the generator.
Precisely. If that pike stays fully open and too much calcium floods in, it triggers a catastrophic structural failure. A literal pore opens in the mitochondrial membrane, releasing toxic proteins that initiate apoptosis.
Which is programmed cell death. The cell just self-destructs.
Right. So the cancer cell has to meticulously fine-tune this GV complex to get, just enough calcium to divide, but not a drop more.
Which perfectly explains the behavior of the PTN protein that the source material mentions.
Yes, PTN is crucial here.
Because in a healthy cell, PTNN acts as a molecular brake on this funnel. Right. It restricts the calcium flow to prevent overgrowth. But in aggressive cancers, the PTN gene is mutated or just deleted entirely.
The brakes are literally cut. And under extreme cellular stress, like say when a tumor is being bombarded by chemotherapy, the cancer actually deploys emergency overrides.
Oh, like the sigma-1 receptor.
Exactly. Normally, the sigma-1 receptor is dormant. But during severe stress, it migrates to the ER, binds directly to the IP3R valve, and just forces it into the perfect open position.
Wow. So it guarantees the mitochondria get that exact surge of calcium to survive the chemo.
Yeah, it's brilliantly insidious. And the cancer is also modifying the physical outer hull of the mitochondria. The review highlights a protein called FUNDC1.
Which is part of the city's sanitation system, right?
Basically, yeah. Usually, FUNDC1 flags damaged mitochondria for a recycling process called mitophagy. It cleans up the broken generators. But in cholangiocarcinoma...
That's a highly lethal bile duct cancer, right?
Yes, very aggressive. In that specific cancer, the tumor actually overexpresses FUNDC1 to constantly repair and optimize its energy grid.
But the clinical implications of that are wild, because researchers found out that if you selectively block FUNDC1 in these tumors, the damaged mitochondria start piling up.
Right, they accumulate and begin leaking these reactive oxygen species.
And that triggers a very specific alternative form of cell death, right? Ferroptosis.
Exactly. Ferroptosis is highly dependent on iron toxicity. So the cancer essentially rusts from the inside out just because its sanitation grid collapses.
It's amazing. And, the context dependency of these bridges is also just wild to me. Take the tethers known as mitofusins, MFN1 and MFN2.
Oh, yeah, the adjustable bungee cords.
Yeah, they act exactly like bungee cords between the ER and mitochondria.
Yeah.
In liver and colorectal cancers, MFN2 behaves like a tumor suppressor.
So if you activate it there, it tightens the gap, forces a calcium overload, and kills the cancer.
Right. But in gastric and lung cancers, MFN2 flips entirely. It acts as an oncogene. It actually loosens the gap to prevent calcium toxicity while somehow promoting tumor invasion.
Which really demonstrates that we can't just develop, like, a universal bridge-breaking drug. We have to understand the specific architectural layout of the tumor we're targeting.
Yeah, there's no magic bullet.
Oh,
So let's take a quick second to recap what we have so far for the listener.
good idea.
The ER and mitochondria are physically fused by that GIV calcium pipe. Cancer survives by maintaining this razor-thin balance of calcium flow, right? Maximizing energy output while actively suppressing the apoptosis and ferroptosis pathways that would normally destroy it.
It's a perfect summary.
So think about this. Imagine you are a pharmacologist trying to drug this system. Would you try to completely sever the connection to starve the energy grid? Or do you design a molecule to jam the valve wide open and intentionally drown the mitochondria in calcium?
Mm-hmm.
Both strategies are actually in clinical trials right now.
It's a fascinating problem.
It really is.
Yeah.
But, a supercharged energy grid generates massive amounts of cellular waste. So how does a tumor manage its recycling and waste disposal systems?
Well, this requires us to trace the ER's connections to endosomes and lysosomes.
The trash cans of the cell.
Exactly. When a cell engulfs material, it forms an endosome. And as this endosome travels deeper into the cell, it matures, it becomes increasingly acidic, and it physically swaps out surface identity tags.
It transitions from a protein marker called Rab5 to Rab7.
Right, and eventually it matures into a full lysosome, which is just packed with highly destructive digestive enzymes.
And the ER acts like this central highway system for this whole maturation process. It physically tethers to these endosomes, guiding where they go.
But cancer heavily manipulates this highway, specifically regarding cholesterol transport. The paper highlights a tethering protein called STARD3.
Okay, so STARD3, how does that work?
So STARD3 is situated on the late endosome. It physically reaches across the cellular void and locks onto a protein called VAP on the ER membrane.
VAP.
Yeah.
So it forms a dedicated cholesterol pipeline.
Exactly. And in breast and prostate cancers, STARD3 is massively upregulated. The ER pumps so much cholesterol into the endosome that the membrane physically stiffens.
And because of that hoarding, the endosome is basically paralyzed, right?
Yeah, it's frozen. It cannot complete the maturation process into a lysosome.
Which sounds like a minor glitch, but the downstream effect is just devastating. Because normally when a growth signal finishes firing, like the HER2 receptor in breast cancer, it gets sucked into a lysosome and digested.
It's the cell's way of turning the switch off.
Right. But if STARD3 prevents the lysosome from ever forming, that HER2 receptor is never destroyed.
Exactly. It remains active on the cell surface, continuously screaming at the cell to divide and conquer. The cancer intentionally sabotages its own recycling plants just to ensure its growth signals are never silenced.
It basically secures immortality by breaking the off switch. But cancer doesn't just halt the lysosomes, right? It actually weaponizes them using a protein called protrudin.
Yes. If lysosomes are the city's demolition vehicles, protrudin is basically the rogue dispatcher.
I love that analogy. It's like cellular battering rams.
Exactly. So protrudin is an ER-resident protein. It latches onto these highly acidic Rab7-tagged lysosomes and simultaneously binds to kinesin 1, which is a cellular motor protein.
So it's grabbing the acid sac and attaching a motor to it.
Right. And protrudin literally drags these toxic payloads away from the center of the cell, hauling them along microtubule tracks all the way to the extreme outer border of the plasma membrane.
And once they hit that cell border, these lysosomes fuse with the outer membrane and just violently eject their acid and enzymes into the surrounding tissue,
dissolving the healthy tissue that's acting as a barrier. And this forms microscopic spike-like structures called invatopodia,
which is the exact mechanism of metastasis in a lot of aggressive breast cancers. The cancer isn't just like passively wandering into the bloodstream.
No, not at all. High levels of protrudin allow the tumor to actively secrete acid, digest a physical hole through the basement membrane, and migrate out.
It's eating its way out. That's why elevated protrudin correlates so strongly with poor patient survival.
It's an active invasion.
So just to give a quick mini recap of this waste management network, by altering tethers like STARD3, the cancer hoards cholesterol to freeze recycling, keeping its growth receptors switched on permanently. And through protrudin, it conscripts these demolition vehicles, dragging them to the borders to blast through tissue.
It completely recontextualizes how we view a spreading tumor, doesn't it? It's a highly coordinated engineering project.
It really does. I mean, think about it. How does this change your view of metastasis? It's actively bringing its digestive system to the front lines. But what happens when these organelles bypass that central ER highway entirely?
Well, then we see direct communication between the mitochondria and lysosomes, and this direct contact is largely governed by that Rab7 identity tag we talked about earlier.
Right, the one on the mature lysosomes.
Exactly. When they dock directly, they are primarily trading two highly volatile currencies, iron and calcium.
So let's look at the iron exchange first. That's mediated by a transport protein called DMT1, right?
Yes, DMT1 acts as a pipeline transferring iron directly from the endosome into the mitochondria. And mitochondria require iron to function. It's essential for the electron transport chain to make ATP.
But iron is highly reactive. The sources detail this wild case study involving a triple-negative breast cancer line, MDA-MB231.
A very aggressive tumor model.
Yeah. And researchers observed that in these tumors, elevated DMT1 levels meant the mitochondria were hoarding iron, resulting in massive energy production and rapid growth.
So the logical therapeutic intervention seems obvious, right? Just silence DMT1, cut off the iron pipeline, starve the mitochondria, and halt the cancer.
But when they tried that, the research showed this horrifying biological backlash.
Yeah, it's wild. When scientists silenced DMT1 to induce iron starvation, the cancer instantly recognized the deficit.
It triggered a panic response. The iron starvation activated a hypoxic or low oxygen survival pathway.
And that pathway rapidly upregulated motility genes. So while the primary tumor's growth might have actually slowed down, the cells themselves became incredibly mobile,
resulting in a massive increase in lung metastases.
Exactly. You block a supply road, and the tumor instantly builds an interstate highway to a new organ.
Which is terrifying. And this adaptability extends to how lysosomes handle calcium, too, using an escape hatch called TRPML1.
Right. We already established that a calcium overload will kill the mitochondria. So to prevent this, cancer utilizes TRPML1 on the lysosomal membrane as a pressure release valve.
So it bleeds off just enough excess calcium directly into the mitochondria.
Exactly. It sustains vital signaling pathways, but carefully regulates the flow to completely inhibit ferroptosis. And this specific TRPML1 mechanism is heavily used to keep cancer stem cells alive.
And those are the cells responsible for tumor recurrence after chemotherapy, right?
Yes, they are the most resilient cells in the tumor.
Wow. Okay, so let's solidify this bypass network. Mitochondria and lysosomes directly dock to manage iron and calcium via DMT1 and TRPML1. This delicate balance prevents that iron-rusting cell death while maximizing energy.
Right.
But manipulating it is dangerous because, as we saw with the DMT1 silencing, depriving a tumor of iron can unintentionally trigger a massive metastatic breakout.
It just invalidates the idea of a single magic bullet. We have to target multiple infrastructure hubs simultaneously.
I mean, if a cancer cell can instantly adapt to being starved of iron by becoming more metastatic, it tells you just how multi-layered our treatments need to be.
Absolutely. Which really brings us to the outer districts of this cellular city. We've mapped the central hubs, but a tumor's true resilience lies in its emergency reserves and border communications.
Yeah, its outer infrastructure is fascinating. I want to do a rapid-fire map of these. Let's look at the nucleus and the mitochondria first.
Okay, so under extreme therapeutic stress, the mitochondria will actually physically migrate and wrap around the nucleus.
Literally hugging it. And a tethering protein called TSPO forms a bridge, delivering cholesterol directly into the nuclear envelope.
And that cholesterol delivery fundamentally alters the fluidity of the nuclear membrane. It clusters specific signaling molecules together, directly sustaining the NFPB pathway.
And NFP is like a master transcription factor.
Right. When it remains active, it constantly reads DNA to produce survival proteins. It effectively hardwires the nucleus to refuse apoptosis, no matter how much chemotherapy is present.
Unbelievable. And then there are the peroxisomes. These are the small organelles responsible for lipid processing. They tether to the ER via a protein called ACBD5.
And the critical enzyme at this contact site is ACSL1, which actively burns fatty acids for alternative energy.
And the role of ACSL1 is just a masterclass in tumor adaptability. In endometrial cancer, if you deplete ACSL1, the tumor cells lose their liquid energy source and rapidly die. Right, there's always a but. If a tumor mutates to drastically upregulate ACSL1, it alters the lipid composition so severely that it triggers EMT epithelial mesenchymal transition.
Right, which is the biological switch where a stationary cell completely changes its physical shape, loses its grip on surrounding cells, and becomes highly mobile.
So hyperburning fatty acids literally provides the fuel and the structural change needed for metastasis.
Exactly. And if the tumor can't burn fat, it relies on lipid droplets. These aren't just inert balls of fat. They are dynamic emergency fuel depots.
Like the cellular pantry.
Exactly. During starvation, like when a tumor outgrows its blood supply, the specific tethering proteins, PLIN5 and FAPP4, physically lock these lipid droplets directly onto the mitochondria,
guaranteeing a zero-loss transfer of fatty acids straight into the mitochondrial furnace. The cancer ensures its energy grid stays online, even in a totally starved environment.
It's brilliant. But the most visually striking mechanism has to be how the ER interacts with the plasma membrane, the absolute outer wall of the cell. This is managed by a protein called STIM1.
Right. And the mechanism here is incredible. When the ER's internal calcium drops to dangerously low levels, STIM1 acts as a physical sensor.
It undergoes a massive shape change. It actually elongates, stretching across the empty space of the cell to reach the outer plasma membrane, and physically latches onto and rips open external calcium channels called ORA1.
It's literally reaching out a window to pull in supplies from the street.
Yeah. This process, store-operated calcium entry, or SOCE, allows the ER to replenish its grid by draining calcium from the extracellular bloodstream.
And in malignancies like cervical cancer, STIM1 becomes hyperactive. That massive influx of calcium alters cellular signaling to drive angiogenesis.
So the tumor actually forces the surrounding tissue to build brand new blood vessels specifically to feed its own mass.
So to contextualize this outer infrastructure, the cancer cell has redundant supply lines for every scenario. If it's stressed, TSPO reinforces the nucleus. If it needs to move, ACSL1 burns fat. If it's starving, lipid droplets plug into the generator. And if it's out of internal voltage, STIM1 reaches outside to steal calcium.
And what makes this nearly impenetrable is that these aren't just two-way streets. The imaging shows three-way and even four-way junctions.
Right, like a group chat. The ER, mitochondria, and plasma membrane can all dock simultaneously to create a unified sensory node,
tracking growth signals from the outside and instantly distributing the metabolic workload.
And this multi-organelle coordination is also how the tumor hides from the immune system, isn't it?
Yes. Cancer cells synthesize a thick, highly complex sugar coating called a glycocalyx. It acts as an invisibility cloak, physically preventing our immune system's T cells from recognizing the malignant proteins underneath.
But constructing this massive cloak requires flawless lipid biosynthesis, which can only happen at these specific three-way ER-mitochondria junctions.
Exactly. But this complex wiring also presents a massive vulnerability. Researchers found that if you chemically disrupt the contact sites between the ER and the endosomes, the sudden break in physical tension acts like a molecular tripwire.
It accidentally activates the NLRP3 inflammasome,
which is essentially an intracellular fire alarm. It immediately alerts the body's innate immune system that something is critically wrong inside that cell,
stripping away the tumor's stealth advantage. So how close are we to actually drugging this physical infrastructure in patients?
We're getting there. We are already finding compounds that manipulate these bridges. The VDC channel, the mitochondrial side of our calcium pipe, is a major target.
Okay.
Interestingly, a synthesized derivative of resveratrol, which is a compound originally found in red grapes, shows remarkable ability to alter these contact sites in cancer models.
It doesn't break the bridge, right? It actually weaponizes our Goldilocks dilemma from earlier.
Yes, exactly. The resveratrol derivative locks onto the complex and forces the calcium funnel wide open. It overrides the cancer's fine-tuning, shoving a massive surge of calcium from the ER into the mitochondria.
So the generator overloads, the membranes rupture, and the cell is forced into immediate apoptosis.
We are using the tumor's own hardwired grid to electrocute it.
That is an incredibly elegant solution. So let's make sure this final picture is clear. The cell uses complex, multi-organelle junctions to manage everything from emergency fuel to immune cloaking. And by targeting these physical bridges, we can either trip an alarm that wakes up the immune system or use drugs to force a lethal calcium overload.
Exactly.
But this raises a really critical hurdle for you, the listener, to consider. Every healthy cell in your heart, your liver, your brain relies on these exact same membrane contact sites to survive.
Yeah, that's the rub.
Right. If we deploy a drug that fundamentally alters the spatial plumbing of the cell, how do we ensure it only collapses the mutated infrastructure of the tumor without shutting off the power grid to the rest of the body?
And that is the defining challenge of next-generation oncology. We are constantly searching for specific structural nuances, subtle mutations in proteins like VAP or RAB7 that only exist in the tumor's contact sites, allowing for highly targeted disruption.
So to synthesize our whole journey through the source material today, we have to stop viewing the cell as a disorganized soup. It is a highly ordered, physically interconnected metropolis. Cancer's greatest superpower isn't just genetic mutation, it is urban rezoning. It hotwires the calcium pipes for limitless power, it paralyzes the cholesterol delivery trucks to keep growth signals firing, and it mobilizes acid-dumping demolition vehicles to violently conquer new tissue.
And understanding that physical architecture, it opens up a deeply provocative future for medicine. If these contact sites rely on specific, physically interlocking proteins to function, we could theoretically design artificial organelles.
Wait, like a molecular Trojan horse?
Exactly. Imagine engineering a synthetic nanoparticle wrapped in a lipid membrane, perfectly mimicking a late endosome.
Okay.
We program its surface with synthetic RAB7 and STARD3 proteins. When injected, the tumor's ER would recognize the tags and physically dock with the synthetic nanoparticle.
Thinking it's just a normal endosome.
Right, but instead of transferring cholesterol, the nanoparticle detonates a microscopic payload, perhaps a calcium flooding agent or an iron chelator.
Just scrambling the tumor's internal communication network entirely from the inside out.
We are transitioning from fighting cancer with the blunt force of traditional chemotherapy to infiltrating and dismantling its infrastructure through microscopic sabotage.
That is mind-blowing. Think about that the next time you look at a diagram of a cell. The spaces between the structures are just as important as the structures themselves. Thank you for joining us on this deep dive into the hidden architecture of cancer. Keep questioning the unseen structures of the world around you, and we'll see you next time.